Hello friends and viewers. Today I am going to talk about the types of vaccine that we have right now in the world, or some vaccine are ongoing. Due to the pandemic of the COVID-19, most of we are very interested to learn about what type of vaccine are going on for the COVID-19. So I have made this video especially to inform all of you who are interested on the vaccine. From the beginning, when Edward Jenner was working with the smallpox vaccine, we have not developed something very special or something exclusive in the vaccination process or the mechanism of vaccine that can induce a very protective immunity. We still rely on the vaccination concept that was developed by the Edward Jenner. From the beginning, we have the live attenuated vaccine. These type of vaccine are prepared mostly by attenuating the live pathogen like the bacteria and the virus. The attenuation is a process that how the virus can be make more weakened compared to the wild type of virus. And this can be done by passing the virus through the tissue culture or the embryonated egg or the live animal. For example, the measles vaccine, mumps vaccine or the rubella vaccine, which are considered as mRNA vaccine, those are live attenuated vaccine. Also the rotavirus vaccine, smallpox vaccine and the chickenpox vaccine, all are live attenuated vaccine. The second type of vaccine is the killed organism, live attenuated vaccine, because they contain the living pathogen. In some point, they can get stronger and can cause disease, especially who have the weakened immune system, like the immunocompromised people or the people having the chemotherapy or the HIV infected people. So, scientists have considered killing all of the bacteria or the virus when they are administered as a vaccine. So they are called the killed vaccine. So they are safe, they cannot replicate inside of the human body. And this type of vaccine like the hepatitis A, flu shots only, and the polio shots only, and the rabies vaccine. The third type of vaccine is considered as a subunit vaccine. The subunit vaccine are a type of vaccine. Those are parts of the whole organism. So that's called the subunit. This type of subunit vaccine can be divided into different types depending on which parts of the virus or the bacteria is taken as a vaccine components. Like the first one is the toxoid vaccine. The toxoid vaccine are prepared from the toxin of the pathogen. When the virus or the bacteria produce the toxin which are pathogenic for the human, they are treated with chemical or heat to make it less toxic. However, it retains the original immunogenicity. That's the term toxoid comes. Toxoid means the toxin part which are chemically treated and non-toxic. When they are given in the vaccine formulation, they are called the toxoid vaccine. This type of vaccine like the diphtheria, pertussis and the tetanus, those are considered as the toxoid vaccine. They are sometimes given all together and called the DPT vaccine. The second subunit vaccine are the conjugate vaccine. When two subunits are conjugated together, they are called the conjugate vaccine. Because polysaccharide, which is a part of a pathogen, sometimes is not immunogenic. So they are normally conjugated with the protein subunit of that pathogen or from the other pathogen. In most of the cases, the polysaccharides are conjugated with the tetanus toxoid. So this type of vaccine like the Haemophilus influenza type B or the Hep B vaccine. The third one is the recombinant protein vaccine. This type of vaccine was introduced when the scientists could recombine two different type of DNA into one. This is called recombinant DNA technology. For example, the hepatitis B vaccine was produced by this technology. So scientists took the DNA part from the hepatitis B virus, which encodes the hepatitis B surface antigen, and those genes are inserted in the yeast, and then let the yeast grow the hepatitis B surface antigen inside of the yeast cell, and then those protein are extracted from the yeast and those are introduced in the formulation of the vaccine. That's how the hepatitis B vaccine was produced by the recombinant DNA technology. The fourth type of vaccine, called the peptide vaccine. This is the concept that we do not need the whole protein to induce the immunity in human. Mainly the T cell use the part of the protein to induce the immunity. So the peptide part is trying to introduce in the vaccine formulation to induce a protective response for the cancer, Alzheimer's disease, to make a peptide vaccine. Now in the market, we don't have any peptide vaccine, but the research is ongoing to establish a new peptide vaccine. So the next type of vaccine is the nucleic acid vaccine. Nucleic acid based vaccine are mainly three types: the DNA vaccine, RNA vaccine, and the vector vaccine. DNA vaccine is straightforward, like you have some DNA and you will administer this DNA into human, and those DNA make specific type of protein. That proteins are immunogenic in nature, so they will give you the immunity of that specific protein after the production of protein inside of the human body. This type of DNA vaccine are also ongoing, so there is no vaccine in the market right now. Mainly the cancer vaccine and the HIV vaccine are considered as the ongoing practice of the DNA vaccine. The second type of vaccine are the RNA vaccine. And if it is RNA vaccine, mostly the mRNA vaccine are considered. And this type of vaccine also ongoing on the market, and especially for the COVID-19, two vaccines are mostly promising right now. So one is by the Pfizer and the second one is by the Moderna. Those two companies are developing mRNA based vaccine, more than 90% safe and efficient on phase 3 trial. So this type of vaccine normally produced by making the mRNA, and this mRNA that can produce the immunogenic protein of that specific virus. For example, for the COVID-19, they have picked the spike protein mRNA to introduce in the vaccine. And the third type of vaccine is the vector vaccines. This type of vaccine are produced by the vector inserted with the DNA. This type of vector are normally the virus vector. Here the vector means that who can carry the DNA inside of the cell, because normally the DNA cannot penetrate inside of the cell, so they need some vector. And these viruses are non-pathogenic, mostly non-replicating inside of the human body, so they are safe. Oxford University and the AstraZeneca has produced a COVID-19 vaccine, that is the vector vaccine, and they have shown more than 90% safe and efficient in phase 3 clinical trial. And here they have used the spike protein DNA inserted into the normal cold producing virus that was isolated from the chimpanzee. So in terms of the virus, this is safe because this is from the chimpanzee, not from the human, so they cannot infect in human. like the human cold virus, and also the spike protein DNA that is introduced inside. They can produce the spike protein. Hope you have understood the basic concept of vaccine and the types of vaccine. If you like the video, thumbs up and subscribe for the next video I'm going to prepare. Thank you for watching.